So, good morning, everyone. My name is Bulisa Masiga, and this is my colleague.
Good morning. My name is Judy Nduta.
And as previously mentioned, we will be doing our presentation on corals and coral restoration. And me and Judy work at an organization called Revolution Foundation. We're based in Shimoni, Kenya. And essentially what we do is coral restoration. So we're basically restoring degraded reefs back into healthy and thriving coral reefs. And we are reef rangers. So as the title suggests, we are basically rangers who are stewards for the coral reefs in and around the Wasini Channel. So our area of work, where the project is located, is quite unique because it's right within in and around the area, the Kisite Mpunguti Marine Park, which is a marine park in Kenya, and it's a vibrant marine park. And it's unique. What's unique about the area is how the marine resources are managed. So Kisite Mpunguti Marine Park is a government organization, and it's a government park that's managed by the government. But the areas and the resources around the park are locally managed by beach management units. And in and around the Wasini Channel, there are 4 beach management units, and we are currently working with one, the Mkwiro Beach Management Unit. And that whole local management system just eases the process of managing all these different resources in different locations, and it helps us manage them together. Okay, so as previously mentioned, our presentation is on corals and coral restoration, and we're going to give you an underwater glimpse of what that's about. So we can't talk about corals or coral restoration without first talking about what a coral is, yeah? And a coral is closely related to sea anemones and jellyfish. And what a coral is basically a small soft-bodied animal and we call that soft-bodied part of the coral the polyp. So we'll look into the physiology of a coral, yeah? And you'll allow me to use a pointer here. So this particular icon will just show what is basically the coral colony, and that's made up of various fragments, yeah? If we look closer into this icon, we see the polyp, and as indicated down here, there are hundreds of polyps that make up a colony. So we'll look at the structure of a coral polyp, and basically a polyp is surrounded by a calcium carbonate skeleton. As we see here, this outline here on the outside is the calcium carbonate skeleton, and it's surrounding the main body of the polyp, which is right in the middle of the skeleton. And the calcium carbonate skeleton is a feature that distinguishes it from close relatives like sea anemones. Polyps have one opening, and that's its mouth, which is surrounded by tentacles. The tentacles have nematocysts, which are stinging cells that enable the coral polyp to catch small organisms. The body of the polyp contains digestive and reproductive organs. So this is a nice illustration of the polyp, and we'll look in a couple of slides. We'll look at a more real-life example of that. So if we zoom in even further, we see the tentacles here surrounding the coral polyp. So this represents the tentacles and this represents the coral polyps. Yeah. So as we said earlier, the stinger cells are located on the tentacles, and these stinger cells are called nematocysts. So most tropical reef-building corals have an additional source of food, which is often the main source of energy. And this additional source of food is facilitated by zooxanthellae, which are microscopic algae that live in symbiosis with corals. They are single-celled photosynthetic organisms that effectively share the food they produce through photosynthesis with the coral polyps. In exchange, the coral polyps offer zooxanthellae nutrients in the form of waste products as a that's produced as a byproduct of digestion, and most importantly, a nicely protected home. This symbiotic relationship gives tropical corals sufficient energy to grow relatively quickly enough to build reefs. Yeah. So I personally think that symbiotic relationship is quite interesting and unique. So here's a more close-up picture of what we just talked about. Yeah. So as we see here, the corallites or the calcium carbonate skeleton surrounds the polyp. Yeah. We see the opening there, which is its mouth, and we see tentacles. If we look very closely, we can see the tentacles actually surrounding the opening. So zooxanthellae are largely responsible for the green, brown, and red colors observed in a large majority of corals. Other colors like blue, purple, violet are a result of the coral polyp itself. Individual polyps are extremely small in comparison to a coral reef, and polyp sizes vary. However, most usually have diameters that fall within the 1 millimeter mark. The genus Fungia has the largest coral polyps that have a diameter of about 12 centimeters. So how do corals reproduce? Corals can either be male or female or hermaphrodites, and they reproduce either sexually or asexually. Sexual reproduction promotes genetic diversity in the reef, while asexual reproduction is usually attributed to increasing the overall size of a colony. Asexual reproduction results in colonies that are genetically identical to each other, and it usually happens in two ways: budding and fragmentation. Budding results when a polyp is formed directly from the parent polyp. So a parent polyp will divide itself in two. So let's talk about fragmentation for a little bit. And as we look at the screen, we have a diver here who's holding a coral fragment. And on the right, we have coral fragments that are lying in coral rubble. So fragmentation can occur intentionally, but is mostly intentional. When it occurs intentionally, a coral will actually disintegrate a specific location on its skeleton, allowing for easier breakage. Unintentional fragmentation, which is more common, occurs when a colony is subjected to physical disturbances such as storms, fish predation, and fishing traps, for example. The chances of survival after fragmentation is largely dependent on the substrate the fragment finds itself on and the fragment size. So corals require a hard substrate to attach to, and once they attach to that hard substrate, they can effectively grow. So now let's talk about sexual reproduction and spawning. So as I said earlier, sexual reproduction results in diversified gene pools. Majority of all hard corals are hermaphrodite, while others have separately sexed individuals, and in some cases, entire colonies. Many corals reproduce sexually through spawning, where eggs and sperm are released into the water column. If we look at our screen here, we see two images, and one image is coral fragments or colonies that are being grown in a controlled environment. And you can see as they spawn, they release the eggs and sperm into that water column. On the right, we have a coral colony that's in a more natural environment. And once again, we see eggs and sperm released into that water column. The gametes will float towards the surface where fertilization occurs, and a planula is formed after successful fertilization. So a planula to corals is essentially what a zygote is to human beings. The result of a successful fertilization is coral recruits. So the planulae will then look for a hard substrate to settle on, where they will develop into an individual polyp. Some planulae can survive for months on end before finding a hard substrate to settle on due to their symbiotic relationship with zooxanthellae. So it's what I find that actually quite interesting that relationship is formed in some corals at such an early stage in its life. And mass spawning events are relatively common, where colonies of varying species synchronize to form one large spawning event. Mass spawning events in the Western Indian Ocean, where we're located, however, are not widely understood, and little is known as to whether they even occur within the region or not. Mass spawning events in the Great Barrier Reef and the Caribbean, however, are well documented and better understood, with factors like temperature, the lunar cycle, and time of day all influencing the release of gametes during their mass spawning events. So we'll just look at the pictures again and talk about them a little bit more. On the left side here, we have a very, very small Porites colony. And as we can see, it's attached firmly to hard substrates along a reef, an existing reef. So what happened here is a planula found itself and attached itself to that particular location. And as you can see, it's thriving and it's starting to grow into its own colony. On the right here, we have two different coral recruits that have attached to a structure underwater. We have one on the left and one on the right. This is a branching coral and this is a massive coral, but both have actually attached to the structure underwater. And as I've said before, that hard substrate allows the coral to firmly attach and to thrive. So how do we move from corals to coral reefs? As previously mentioned, most corals live in colonies. So the size of a colony tends to be much larger than the polyps making up the colony. I think this is a pretty cool picture. It was taken in the Wasini channel of a coral reef completely exposed, almost completely exposed during a low spring tide. And what this picture shows is like all these different colonies that are actually made up of, I'd say, thousands and thousands of polyps, individual polyps. These are some more pictures depicting that, just different reefs. So when you look at a reef, there are all these little tiny animals that actually make up the reef. And if we look at this first picture here, we can see that they're totally different species, a number of different varying species that will make up that reef. Another picture here was taken during a low spring tide within the channel. But even just this small area here can show about 4 different species of coral. So each of these are made up by thousands of little animals that were called polyps. So there are 4 types of coral reefs, and this image, as the screen shows, all 4 types are displayed here. So we have fringing reefs, in the first image, and they grow along coastlines, and they tend to be separated from the shore by shallow lagoons. On the right here, we have a barrier reef, and unlike fringing reefs, barrier reefs are characterized by a deep, widespread lagoon separating the reef from the shore. Barrier reefs consist of various reefs combining and forming a vast barrier along the coastline. The largest coral reef is the Great Barrier Reef that extends for more than 2,000 kilometers. This image here shows an atoll reef, and an atoll reef is formed when an underwater volcano on an island slowly sinks and a reef starts to form around its perimeter. On the right here, we have patch reefs, which grow, as the name suggests, grow in patches. Another image that was taken during a low spring tide in the Wasini Channel, and it shows like one patch of reef here and another patch of reef here. Patch reefs usually are found within lagoons created by barrier reefs or atoll reefs. So let's talk about the biodiversity that comes about because of coral reefs. And coral reefs are in fact the most biodiverse marine ecosystems. An estimated 25% of all marine species find their food or shelter in coral reefs. An estimated 7,000 species of fish, invertebrates, plants, turtles, birds, and marine mammals are supported by coral reefs in terms of food and or shelter. And here's some more images just displaying that biodiversity. We have fish here, we have a mammal, a marine mammal here, and we have a reptile here. They're often compared to rainforests regarding their biodiversity. It sometimes comes as a surprise that coral reefs actually only occupy about 1% of the ocean floor, and yet they display so much biodiversity. Coral reefs bring about various socio-economic benefits. So in addition to their ecological prowess, coral reefs also have immense socio-economic value, providing food, income from tourism, and shoreline protection. Recently, there have been drug developments, and basically there's research going into how you can make medicines for certain organisms that are found within coral reefs. And the first picture, we see a fisherman here with a healthy catch from the deep sea. And the middle picture shows what it looks like, what a basket trap looks like from the inside. And on the right here, another reef that was exposed during a low spring tide, and basically depicts what happens as the waves coming into shore come into contact with that reef. Here's some more images. This is a fisherman. This is a restaurant right across the Wasini Channel. And this is more pictures displaying tourism. And all these pictures are taken in and around the Wasini Channel. So these are clear local examples of how the coral reefs are benefiting the communities and us in and around Wasini Channel. Unfortunately, due to various anthropogenic activities like overfishing, destructive fishing methods, pollution, global warming, and the introduction of invasive species, among others, coral reefs are under constant threat of degradation and are constantly being pushed past thresholds that they can bear. So we're going to look at the threats here, and I've divided them into global threats and more localized threats. And the main global threats affecting our coral reefs come about because of global warming and ocean acidification. And the result is bleaching events, which I'm sure most of us are familiar with. And the main way and most effective way we can tackle this is by trying to make coral reefs as resilient as possible, yeah, so they can withstand these threats. And we look at a few localized threats here, and we have unsustainable tourism or recreational activities. In this image here, we have these are divers, and they're standing, resting, posing on the reef. And what this does, it actually breaks the coral fragments into tiny, tiny fragments, reducing the chances of survival for those particular reefs. We have anchors here, and once an anchor attaches or is latched onto a coral reef, when it's pulled or it drags, it destroys the reef. An alternative to anchors, which is very common here in and around the Wasini Channel, is using mooring lines, which are a very effective alternative to anchors. We have ghost nets, and ghost nets are essentially nets that are lost at sea. Usually these nets can get entangled in around the corals and around coral colonies, and they restrict their growth when they completely enclose and surround a coral reef. We have pollution, which is not limited to plastic pollution. It also includes oil spills, agricultural pollution, industrial pollution. And in this case here, we see a sack, presumably made out of sisal, I think, or it could be plastic, but we see the sack that's that's completely covered a particular fragment. And in the last image here, we talk about unsustainable fishing. Once again, this particular image is displaying what the remains of blast fishing activities looks like, but there are other examples of unsustainable fishing that are not exactly limited to blast fishing. Here in the Wasini Channel, the reefs were heavily degraded due to blast fishing in the past. I think specifically in the 90s. So fishermen would use dynamite to actually blow up the reefs and collect the dead fish after. So a lot of the areas we're restoring look like this. And with that, I will hand you over to Judy, who will take us through the next phase of the presentation, and that will give you a clearer view into what we do here at Revolution and our coral restoration.
So thank you again for having us and joining us on this presentation. So my name is Judy, and I'll be taking you through the restoration part of the presentation. So background information about our organization. So Revolution Foundation, as the name suggests, is characterized on the name reef. So what you're doing here, trying to restore the reef. So we are an NGO that's been operating since 2015. So we work with the community, with the local community around here. We work closely with the Quiro Beach Management Unit that has the mandate of being in charge of this channel, and they are in charge of any conservation or any activity that is going around in that area. So we'll go right direct into the restoration part. So with the restoration, what coral restoration, when we say what coral restoration is, it's basically trying to assist the recovery of an ecosystem that has been destroyed or damaged. So we look at the next slide that's showing a healthy reef and a degraded reef. So what you're trying to do, as Bulisa already said, our area in the previous years was used, the fishermen used dynamite fishing and blast fishing. It's no longer being used, but our area is characterized, it's rubble based. It doesn't have the hard substrate that's needed for the coral to attach itself. So we are trying to bring such an ecosystem back to assisting it to such an area, a healthy reef. Because when you talk about a healthy reef, This is a reef that has a lot of fish, that has a lot of hard corals, that has a lot of sponges, that has clear water, basically an ecosystem that's healthy for a reef to thrive. So when you look at this other picture here, this is an area that's been overgrown by macroalgae. So this happens when we remove herbivore fish like the rabbit fish or the sturgeon fish from the ecosystem. So this herbivore fish feed on the macroalgae. So when we remove them from the ecosystem, the macroalgae overgrows on these coral reefs, and it's not healthy for the reef to thrive when an area is like this. So I'll show you an example of what happens when we remove the herbivore fish and the carnivore fish. When you talk about carnivore fish, we talk about like the triggerfish. So the triggerfish feed on the urchins. So when we remove them from the ecosystem, we have an urchin explosion, and the urchins really feed very, very fast on an area, like we can see here. So this is what happens when we remove fish from the, when there's less balance of fish in the ecosystem. So I'll show you the next slide that is showing our area. So on this side, we're very fortunate. We are close to the City Mfungwiti Marine Protected Area. And so we are here, we're based in Shimoni. Across us, we have the Wasini Channel over here, and this is our area. So this is where we do the restoration, this red part here. So we in collaboration with the Mkwiro BMU. So this is where we put our structures. So on this picture, we can see a closer view of our restoration area here. So this is Shimoni. So here we have the red part is the fish breeding area. So the idea of having this area is to ensure that fish have time to breed and to increase in populations. Because the goal of having our restoration was to restore the degraded area, to help the fisheries increase, the fisheries, and to increase the fish population in this area. So this is the locally managed area, as you can see, and this is the fishing zone. So the fishermen are allowed to fish in this area, but using sustainable fishing practices like basket trap or hard hand lines that are not harming or destroying this ecosystem that you're trying to recover or assist the recovery. So when you talk about coral restoration is a global, or it's big. It's a big. I'll say it's huge. So I'll just majorly explain what you do, the restoration, the method that you use to restore the area, because the restoration methods differ with different areas. So a different area may use a different restoration procedure compared to another area. So given that our area doesn't have the hard substrate for the coral to attach, we chose the method that's best for us, which is coral gardening and using artificial. So when I say coral gardening, I mean growing corals that have been taken into an intermediate phase. In this case, using a nursery phase. So that's what I'll explain. So it involves 3 stages. The first stage is collecting the corals. So these are the corals that have been broken by waves and currents or other unsustainable methods like anchors. So when we have these broken pieces, we collect them and then we resize them into about 5 to 6 centimeters, and then we take them to a nursery phase. So this nursery phase, we attach these corals into these are fishing lines. So we attach these corals here, and then we give these corals a period of about 6 to 12 months, depending on the coral species. So the idea of having a nursery is to ensure these corals have optimum condition for them to grow. So they have sufficient light coming to them, they have clear waters, they have water movement that is bringing the nutrients to the corals. There's less chances of sedimentation because they are higher from the ground. So this is a tree that is naturally, it's buoyant. So this buoy helps it to be stable on itself. So even when the waves and currents come, the the tree in itself is very buoyant. So after having this nursery stage, the corals now are huge. So we take them, we harvest them from this nursery, and then we put them on the artificial reef. So as Bulisa said, the corals need a hard substrate for them to attach themselves and to grow. So we have different designs of structures that we put the corals to grow on. So I'll show an example of a tree or coral tree nursery. So this is how it looks like. It looks like after the corals grow for about a period of time. So they attract fish, as you can see on this picture, and this is a mini tree. So this is a smaller version of a coral tree nursery. So mostly we use these mini trees for research and other experiments being carried out around here. So at this stage, we do a lot of maintenance to ensure that there is no overgrowing of the macroalgae on these loops and on these structures. We clean regularly to ensure there's no overgrowing of the macroalgae. But a study shown done by one of our project leaders, they have shown that it's not necessary to clean as regularly as we used to do, because the fish helps with the cleaning. And we also do the maintenance to ensure that these trees are well taken care of, and they are well in this ecosystem. They're not being moved, they're not broken, to ensure sustainability of this nursery phase. So we can see more fish here. They attract different fish species on the nursery. So as I said, we move these structures onto the artificial reefs. So this is I'll show the different designs of the artificial reef that's used here. So we have an example of a bottle reef. So these are wine bottles in itself, and this is concrete. So we attach the corals on the mouth of these bottle reefs, holding them with a cable tie or a tie wrap to hold the corals here. This is a cage. We attach the corals here on the intersection of this metal bars, and a dome is a bigger version of this cage. And this layered cake, it's basically concrete, but imitating a cake in itself. So the idea of having these different structures and different designs is because different structures attract different fish species and communities. So like a layered cake is a very good example. This layered cake is a very good hiding area for stingrays, for porcupine fish, for eels. A lot of eels hide under these cakes. And these bottle reefs, when they're put together, they attract mostly juvenile fish, and the cages and domes attract different species, different adult and juvenile fish. So we put them together and in what we call a patch. So a patch is composed of all these together in an area, and then they attract different species in the area that you're trying to restore. So I'll show what the actual artificial reefs look underwater. So this is how they look underwater. So you can clearly see the bottle reef, the cakes, and the cages. So this is one of our reef rangers attaching the corals on this metal bar, what we call a cage. So this is a closer look up on what the corals look underwater. You can clearly see after the corals being planted, after a period of time, they start fusing together and forming the reef. And now we are trying to form now. So you can see they attach together, they attract different species. We have more pictures showing the same. You can see how beautiful this looks when the corals attach together and they attract. You can see here a lot of jacks. These are very healthy for the coral reef ecosystem. So yes, we can see another picture showing a comparison between a structure that were placed in 2019, and within a period of about 1 year or 2 years, you can clearly see the difference here. So here we can see there's less fish compared to here. We can see the corals are actually doing really well compared within this period of this period of time. So you can clearly see how well the corals are thriving and how well they are fusing together. So that's what we are trying to do, to see. When we talk about the outplanting process, after we outplanted these corals on these structures, we leave them to grow on themselves. But we do the monitoring part to ensure that we see how well these corals are doing, how the biodiversity is changing, how if there's to ensure the monitoring to ensure to see how well these corals are doing after outplanting them. But we let them grow on themselves. So I'll show a video showing how in the water they look, how these corals look underwater. So that shows how these are corals that were planted a few years ago, and that's how well they're doing in the channel. So I'll show another picture showing another different design of structures. So we can see we have another different design. So with this design, they improve with time, and we try to use different design to see what works best with this area. And we use different species. We have this acropora, we have millepora, we use different species to see which works best and which one is thriving more on this area. So when we talk about all this restoration, we do face some challenges with this with our work. So the challenges that we face, first of all, for restoration to be successful, you need to try and reduce or remove the threats that are causing the decline of the corals. So in our area, we have the threat that is causing the decline is blast fishing. So you have to cap that first so that the restoration to be successful. That being said, we face some challenges with the restoration. So one of the challenges is in this channel that we do the restoration, fishing is allowed. Fishermen can fish in this area, but sometimes the basket trap that they used to fish, they sometimes place them on top of these corals, or sometimes they'll be pushed and come on top of this coral. So when this basket trap is on the corals, it's preventing it from getting all the necessary conditions for it to thrive. Another challenge is, like we can see on this picture, ghost nets. So when fishermen put nets trying to catch the fish, they will be moved. They may be moved by waves of currents and get themselves on our structures. So this is an example of a ghost net on our structures. But we regularly go and check on our structures to remove such cases. Another challenge with the restoration is coral restoration is very costly, considering the research that is going behind the restoration, the science behind the restoration, and the labor behind the restoration. Because you need to monitor, you need to keep up with the research that is going on, and you need the money to build the structures and to keep up with the different designs. So that being said, with the challenges, I'll conclude with some of the benefits that we get from the restoration, because there are very many benefits from the restoration. One of which is the increased genetic diversity and potential for adaptation. So we use different species and different coral genuses and the species level to increase their diversity. So another one is preventing the extinction of some of these species. So with the threats that are going on, the global and the local threats, some species may go extinct. But when we bring about the restoration part of it, we prevent some of these species from reducing or going extinct. So assisting the species migration to new locations, as already mentioned. So when we collect the corals, broken pieces from the coral reef, we move them to different areas and trying to see what area does best for what coral. So yes, and continuing to provide critical ecosystem services. So when the coral thrives, it brings about the biodiversity aspect of it, it brings about the tourism aspect of it, and all the services that are brought about by the restoration itself. So yes, to conclude my presentation, I'll show several pictures, a picture rather, showing some of the people or some of the reef rangers that Bulisa explained that help with the restoration part of it. Because the restoration is not entirely underwater, there's also on land restoration, on land part of it, like the building of the structures, the science behind it that's going on around it. So these are some of people that are involved in the restoration aspect of it. So yes, yeah, Bulisa.
Thank you, Judy. And before we conclude, we'd also like to really appreciate a couple of our colleagues, Evout, Noesta, and Tony Wild for the pictures, the beautiful pictures that you've seen here. And we have to also appreciate Guido Pap for some of the graphics that we used in this presentation. But thank you for listening in, and we hope you enjoyed the presentation.
Thank you so much.
So I'm going to open the floor for the questions, but there's already a question in the chat from Gina. And she's asking how many different species can be found in coral reefs? And have you ever tried to identify the most likely species to find in an artificial coral reef than a natural one, and vice versa?
Coral, when you talk about coral species, there are very many different species. So we have a total of 229 different species from 72 coral genera. So that's a very huge coral species. But when you talk about our area, we have lesser numbers of the coral species, but they're there, they're very many. What's with this? The other part of the question?
The second part, are you able to open the chat, or I can reread it for you?
Okay.
Have you ever tried to identify the most likely species to find in an artificial coral reef compared to a natural one, and vice versa?
So I think, so when we're talking about different species, we're talking about benthic and fish species. So benthic are basically the organisms that are not fish. Yeah. So I can speak on fish species right now. So we regularly conduct both benthic and fish surveys here within the channel at our artificial reefs and compared to natural reefs. And recently there was a student here who concluded a master's thesis on fish biodiversity, comparing both natural reefs and artificial reefs. And from his study, he found out that for the most part, both artificial reefs and natural reefs had a similar amount and similar diversity. And in some cases, the artificial reefs actually had more fish species than the natural reefs.
That's really interesting.
There are benthic surveys that have been carried out recently as well, but I can't speak on that because the results haven't yet been shared with us yet. So when we're talking about sea urchins, sea cucumbers, flatworms, nudibranchs, we don't have the current numbers for those right now.
Okay, no, but I think you answered the question. Thank you so much. Just on that, I've seen on the website, Reefolution.
Yeah.
I saw on your website that there's 3 different projects there. There's the diving, there's the VR experience. So you're speaking about a master student that was there. So if there is anybody that wants to study there, do we approach you or do you share projects that's available? Do you know? Are you able to answer?
What?
So basically, so at Revolution, we work in collaboration, and we're heavily supported by a university in the Netherlands. That's Wageningen University. So a lot of the students who've been coming in recent times have come from Wageningen University. Yeah. But there's also collaborations and partnerships, I think, with Kenyatta University as well. And we're looking forward to expanding those partnerships and collaborations. So at the moment, I think any students who are doing the master's thesis within the project usually come from either one of those two universities.
Okay, thank you so much. Okay, before we go back to the chat questions, there's a question from Mr. Ruan Schlebusch.
There we go. Thank you.
There you are.
Thank you very much for a very informative talk and with beautiful pictures, as Aisha said. I was wondering, when you start, and I hope propagate is the right word here, can you propagate any of or farm with all of the species, or are there species that you focus on? Corals now specifically.
So when you talk about propagating the corals, we work mainly with the corals that we have here. We have, so we have the corals that do best here are the branching corals compared to the massive corals. But different corals work different areas, because we have areas that massive corals do best. But when we talk about our area, the branching corals do best compared to the massive ones. So we focus mostly on the branching corals.
Thank you very much.
Thank you, Ruan.
I think it's also worthwhile to note that as the hope is that as we restore the artificial reefs, that will also attract and give suitable environments for massive corals to thrive as well. I don't know if you can recall from the presentation when I was showing the coral recruits, one of the pictures had two different recruits. It had a branching one and a massive coral that was attached to the structure. Yeah.
Thank you. I'm going to go back to the chat again. You guys got a whole lot of compliments for a very excellent talk. And then there's a question from Gina. No, I read that one. There's one from Claudia Roth. Are there some corals that are more resilient regarding coral bleaching? Because she saw a report on working on species that's resilient to coral bleaching. And then thank you for the talk.
Thank you so much. So when we talk about there are some corals that are more resilient. So corals on themselves, when we talk about massive corals, they are more resilient to coral bleaching. But generally, all corals they take time to grow. So I don't think there are more. I don't know, Bulisa, did you answer that?
I'd say some corals are definitely more resilient, and like for us here in the channel, there are certain experiments that currently run, trying to identify what species are more resilient. So there are numerous experiments that try to tackle various components of the reef and certain conditions that would be affecting the reef. So I can talk about a depth gradient experiment that actually measured, that had corals growing at different depths. So that was trying to see the effect of temperature on the corals at varying depths. And we've also had experiments that are looking at various species and how bleaching affects them. I think we had the most recent bleaching event was in 2021. So even then we had corals, and we actually even actively monitor the water temperatures, and we can identify when it passes a certain threshold. I think in the Wasini Channel, that threshold is 30 degrees Celsius. So once it passes that threshold, the corals start bleaching. So as we continuously monitor the temperatures, we can actually monitor the corals as a bleaching event is happening.
Wow, that's amazing. On that, like, how long does it take for those corals to grow? Because you showed a picture of 2 years, a separation of 2 years. So, like, how long does it take before they're ready?
Corals do take time to grow, and that's why the restoration takes time for itself to be successful on a global scale. But also different species take different times to grow on themselves. So when we talk about branching corals, they are more faster in growing compared to the massive ones. But you can't really say like there's a timeline that the corals have themselves take to grow, but they take time.
Yeah. And when they do, when they are mature enough to be relocated, like, how do you guys know where to place them?
So we have different areas and different locations for them to with different locations that we do the outplanting. So we have what we call the community managed area that we put our structures. So different species, we don't mix the species. So when we take one species, we put them on an area, and then we put another species on another area, but we encourage the diversity of this species.
Yeah. Okay. Then there's another question from, where did I see it now? Mike Lavelle. And it's a very important question. I think on everyone's mind. Firstly, give strength and fortitude to the team, because you guys are doing an amazing job. And it's very informative and encouraging that teams like this are so courageous and dedicated, which is very true. And then the important question is, are visitors and non-students allowed into Wasini Island?
Yes.
Yes.
So, yeah, you're welcome. So actually, we're located on Shimoni. So Wasini Island is across us.
Yeah.
And yeah, visitors are definitely very welcome. I see in the chat box, our colleague Joshua has attached another colleague's email address. So if you're interested in, I say, research and other opportunities, you can reach out to the contact that's been shared there. But we're very, very welcoming, and everyone's welcome to visit. Yeah.
You can just come and see our project and learn more about what we're doing here.
On site.
Yeah, on site.
Oh no, definitely.
So there's a question here that I think that was missed.
Yeah.
Miki Mwamuye. Yeah.
Yeah, I don't see it. Oh, which methods are used to identify the coral species?
Yeah. So identifying coral species is tricky business. But here at Revolution, we are constantly learning different species down to genus and species level. So we have a lot of members of the team who actually know how to effectively identify coral species, especially down to the genus level. As I said, species is very tricky business, but if we come together like collectively, we can try and effectively identify certain coral species. So to answer that question, I guess it would just be educating and like trying to stay ahead of the curve as far as identifying species is concerned.
Yeah.
But we have internal certifications that we do, internal examinations, to ensure that we know exactly what species we're coming into contact with when we're doing our restoration work.
So is it things like the color and maybe the length that it grows to, like the morphology of it?
Yeah, okay. The general morphology of the coral.
All right. And do coral species have subspecies? That's from Aziza Zura.
Yes, corals do have subspecies. We have, when you start from the genus level, we can go down to the species and then subspecies. So they are there. They are subspecies to the corals.
Sure.
Yeah.
So that makes it even trickier to identify.
But it's very interesting when you actually try and learn and understand how the corals are divided.
Yeah,
Yeah.
I can imagine.
Yeah.
Then I think this is also a very vital question. This is from Cindy Saru Churonga. Firstly, she says that was very informative.
Thank
So
you.
far, how many hectares have been restored? And then how can we support the project?
Thank you for that question. And thank you for the compliments. So far, we've restored about 3,500 meters squared. So that's how much artificial reef we have currently.
Wow.
I don't know how I'm gonna combine that into hectares.
Yeah.
But I think that's around, it should be around 3 hectares, is it?
Sure.
I hope I have got the measurements right, but I can confidently say around 3,500 meters squared of artificial reef. And we continue to restore more every other day. And how can you support the project? You can visit the website and follow. I think you can donate to the project, and the instructions for regarding donations are on the website, which I think was attached earlier in the chat box. But
Yeah.
Yeah.
there we are.
Yeah.
So feel free to visit the website and learn more, and hopefully also donate to the project. But yeah, thank you.
No, definitely.
So I see a figure there. So we've 0.35 hectares of artificial reef.
Okay,
So that's one of our colleagues.
We did the calculation. Thank you, Yvonne. Okay, so Johan.
Thank you very much. Thank you for a brilliant presentation. You took us on a dive of your underwater garden, and it was absolutely magic. Thank you.
Thank
you.